Be right back with you, Dr. Clarke. Thank you.
It's not easy. You can't see the molecules, and so we need tricks to look at. And some organisms make these tricks easier than others. And for example, one organism we love are mice, because the technology's been developed that we can go in and basically at will take out a single gene. So to go back to the car analogy, if we want to understand how a car works, and we want to understand what a part does, we take out the part and we let people drive around the car and we see how well it works. And some of that works, some of that's easy. And so if we have a part that we think is involved in aging, we take it out. So the first protein repair gene we found, we took it out of mice. And we were thinking might be a tough experiment because we've worked on this project for 10 years already. What if nothing happens? Because organisms are smart and they can sometimes, if you take out one thing, something else will compensate. But these mice dropped dead. And they dropped dead at 60 days rather than 600 days.
Wow.
And so we said protein repair is important. And what we had hoped was they might do other components of human aging, and so we kid around, like, they start waddling around, they get frail, they turn gray, they talk stupid to their children. In fact, these mice didn't do those things. These mice actually died of seizures. And so we actually have some new biochemistry working here of why would the lack of protein repair cause seizures. Now, certainly that's possible. Seizures are basically when our neuron firings get a little bit uncontrolled, and you have so much neuron firing in there, it's amazing. We don't have that happening more often, okay? But we control it, and if there's more protein damage, we don't control it, okay? And so we're trying to figure out those mechanisms. And in looking at that, one thing we found that was very interesting is that the mice that lack this protein repair have turned on insulin signaling. Now, I want to turn to insulin signaling because insulin signaling is crucial in aging, and there were many talks at the meeting looking at the insulin signal, and also is crucial in
diabetes,
health, and especially in aging and type 2 diabetes.
Correct.
Okay, and it turns out that insulin is a signal that there's lots of glucose in the blood. So it's basically a signal of feast time. Okay,
wow.
Lots to eat. And it may be a signal of sloppiness, for molecular sloppiness, because it's saying it's feast time, let's get this into fat, let's not worry about errors. And so in fact, what we and others have found is that if you turn down this insulin signaling pathway, animals such as worms and flies live longer because they're going in the slow route. So that becomes a question for humans. Can we turn that down either with a drug or by caloric restriction, which is painful but possible, and turn that down and live longer?
And so, one of the things you did mention was Leonard Garante again, and he was working with Dr. Cynthia Kenyon, we believe, and she was one of the first ones that was doing the worm research,
right.
Wasn't she so?
That's right.
Do you guys talk together about these things?
Yeah, and it's very useful because you have different interpretations of the same data. And what I'm saying now is my sort of what I came from this meeting, because the most important thing going to the meeting is getting a new sort of idea set. Okay, you do experiments to test hypotheses, and if you keep your same hypotheses, you're not going to go anywhere.
Do you have some new collaborative relationships?
And we certainly have lots of new collaborative relationships. I'll get an email from someone, Steve, what do you think about this? And I'll say, I know nothing about that, I can't help you. Or sometimes I say, hey, have you thought about this? Maybe our lab could do this, your lab could do this, and we could solve that problem.
That's
and probably,
wonderful.
The majority of our work now is in fact collaborative. And you never know where the next advance is going to come from.
Larry.
Dr. Clarke, we thank you very much for being with us this morning. Question for you. You mentioned the lab has been here 30 years. How long have you actually been involved in this research?
That's how long I've been here. So
you've been here since day one, right out of school?
I came to UCLA 30 years ago. I was a postdoc with Dan Koshland at the University of California at Berkeley, and was hired as an assistant professor at UCLA. And I came here, I started this laboratory, which is still functioning.
Well, earlier you indicated you've got about a dozen people working for you in the lab. What recommendations do you have for new students that want to enter this field?
I think students that enter fields of science have got to be curious, and they've got to be hardworking, and they've got to be able to take a little bit of defeat in light of a a light at the end of the tunnel. Because science is tough. You have to know a lot, and you have to know more and more. And in fact, for aging scientists, one of the things is keeping up with the field, right? As these changes come in, especially changes in computer technology that seems like only the 16-year-olds can understand, right? You've got to get up to that level. At the same time, the 16-year-olds have to learn sort of the most important things that we can teach them. And what's crucial in teaching is that I had a certain knowledge of biochemistry. If I impart that same knowledge of biochemistry to my students, I'm not doing them any services because things have changed. I have to every day say, what's important to teach, okay? What is the most important information to pass on to that next generation that gives them the foundation for them to say, hey, Steve, I've got a new idea, and this is what I want to do now. And they have to have the energy to jump up and do that. And it's exciting. You see students come in, and yeah, some students find, not for me. Other students say, there's nowhere else I want to be than a laboratory discovering things.
You know, it's interesting because I come out of the high-tech world of electronics, and you work in a skunk work somewhere, and all of a sudden at the end of the day, the end of 5 years, you see your product. And as you point out, you really have to have a recognition that you may not see it tomorrow.
Well, it's probably very similar because, you know, sometimes if you're in a skunk works, you don't see a product in 5 years, okay?
That's very true.
And sometimes you work and you don't see something. And so I tell students, you have to enjoy the process.
One of the things I was going to ask you is, and a great question, Larry, but the issue is that if you're interested in coming into this field, do you have to have an extensive chemistry background or biology background, or where would their preliminary studies go and to lead them into your research facility?
Certainly a background helps, but is not essential, okay? You get very bright people, and Seymour Benzer often said, you know, it's great not knowing much about a field because you can come in and you can ask the stupid questions, and the stupid questions are the ones that probably haven't been thought of and may change the whole field. And so having people come in with different backgrounds is fine, okay? If they're bright enough to say, hey, this makes no sense to me. And that's the great thing, students come in. A good student will come in, I'll tell them something, I'll challenge it. They'll say, that doesn't compute, okay? And then I have to rethink that and see if that does compute. And so that whole process then of going back and forth is what keeps me young and keeps my job exciting.
You know, it's interesting because earlier you were mentioning all the different labs that are here, and I was going to ask what school or labs should they consider. But then again, I think it depends on what field they would like to pursue because you have such a variety of options here.
Yeah. Well, you know, for graduate students coming into a program that might have from a dozen to perhaps a dozen faculty, they need to choose a thesis project. And how do you do that? And what we have is we have rotations where we let students spend 10 weeks in a laboratory and get to know the people and hopefully get to know the people in other laboratories and find out what sort of styles they have. You know, I think for many scientists, you could really pluck them in any laboratory. And often in times where that happens, during World War II, you know, basically you took the scientific, you know, elite and you basically put them on wartime problems. And some of these people, you know, they went into something different, they did it, they got excited about it, and they continued in that field. Right?
One of the things I was going to ask you is that Larry was looking for some of your writings, you know, and trying to find out what you, and you've written these giant research books that are just like, I don't know, an encyclopedia practically. So tell us about some of the writing that you do and the research that you do and you put into print.
Well, writing is absolutely crucial because basically it's the condensation. You know, you'll do work and you'll have tons of raw data that basically you may understand, but not too many people are going to understand. And what you have to do is say, okay, from this raw data, what can I say about nature? Okay, what conclusions can I make? And you always try to make the best conclusion. So if you have a story, you say, okay, you know, you make the most outrageous hypothesis you can. Okay, why I've cured cancer today. And then you look at your data and say, well, does it really show that? You say, well, not quite. Then you make the next most outrageous hypothesis you can. And what you're trying to do is to really push the field as far as you can and support it.
Push the envelope.
Yeah. But the supporting is very, very important. I teach a course here in research integrity. And this is something that's NIH mandated, but it's a great idea. And it's a great idea from NIH's point of view is that there's a lot of taxpayer funds that fund research. And the taxpayers are basically entitled to getting a good product for it, okay, where the conclusions are right. How do you know your conclusions are right? And that's where data analysis and statistics are very important.